Welcome to this quick tutorial on what SNARE proteins are and how they function. SNARE proteins are soluble NSF attachment receptors which consist of 100 to 300 amino acids. The main role of SNARE proteins is to mediate membrane fusion events. All SNARE proteins have a transmembrane domain, which is a region that allows the SNARE to interact with lipids or integral proteins on a vesicle or target membrane. Usually there is one V-SNARE located on a vesicle and 3 T-SNAREs located on the target membrane. V-SNAREs have their C-terminus embedded in the target membrane, therefore they are called tail-anchored SNARE proteins. V-SNAREs consist of one chain and cause the vesicle membrane to bulge and distend. Examples of V-SNAREs include synaptobrevin and synaptotagmin. There is always one heavy T-SNARE and 2 light T-SNAREs located on the target membrane. The heavy chain T-SNAREs consist of one chain and belong to the syntaxin family. The first light chain T-SNARE consists of one chain, has 2 SNARE motifs and is often a dimeric molecule, whereas the second light chain consists of 2 chains, one SNARE motif per chain, and is often a trimeric molecule. Examples of T-SNAREs are SNAP25 and syntaxin. So this diagram here shows that SNARE proteins push water molecules away to allow the formation of a hemifusion stalk. To achieve this, an inverted micelle forms with the hydrophilic regions pointing inwards and the hydrophobic regions pointing outwards to make the formation of a hemifusion stalk easier. This process occurs within a microsecond of the membranes coming together. The lipids derived from each bilayer contact each other to form a new bilayer, and rupture of the new bilayer completes the fusion reaction. So, the role of SNAREs in synaptic transmission. First of all, the membrane is depolarized by an action potential, causing the release of intracellular calcium. Calcium binds to synaptotagmin on the vesicle, inducing an affinity of the vesicle to the phospholipid presynaptic membrane. Synaptotagmin binds to syntaxin, whereas synaptobrevin binds to SNAP25 to allow docking of the vesicle to the presynaptic membrane. So just to be clear here, that synaptotagmin on the vesicle containing the neurotransmitter that needs to be exocytosed binds to syntaxin, which is a T-SNARE, on the target membrane, whereas synaptobrevin is a V-SNARE on the vesicle, which binds SNAP25 on the target membrane. And again, this allows the docking of the vesicle to the presynaptic membrane. So an alternative hypothesis to the V-SNARE T-SNARE hypothesis is the Q-SNARE R-SNARE hypothesis. The Q and R SNAREs work in a complex of 3 Q SNAREs and 1 R SNARE. There is usually a glutamine or arginine residue located in the centre of the structural domain. Q SNAREs have a glutamine residue in the centre of the structural domain, whereas R SNAREs have an arginine residue located in the centre of its structural domain. There are 3 different groups of Q SNAREs: QA, QB, and QC, just circled here. Therefore, there are 4 different types of SNARE. So it's QA, QB, QC, and R. The syntaxin-like heavy chains are like QA SNAREs. The light chain SNAREs are like QB and QC SNAREs. Most V SNAREs are like R SNAREs, whereas most T SNAREs are Q SNAREs. Hopefully that makes some sense. So NSF and SNAP proteins, these facilitate SNARE dissociation. So NSF is an ATPase which uses ATP to induce a conformational change and twist SNAP proteins. NSFs attach to SNAP proteins which attach to the cis-SNARE complex and initiate unwinding of the SNARE complex to allow the SNARE proteins to be recycled. Sorry, I just write here for reference that NSF and SNAPs facilitate SNARE dissociation. Just make that nice and clear for you. So again, the NSFs attach to the SNAP proteins, which attach to the cis-SNARE complex, initiates the unwinding of the SNARE complex to allow the SNARE proteins to be recycled. The cis complex is bound by 3 SNAPs and 1 NSF to form a 20S complex. And that concludes this quick tutorial on SNARE proteins. Thank you for listening.